Hello, people of science. Let's talk about mutations and walk you through that worksheet you have. Simply put, a mutation is a change in DNA. Here's my original strand, here's the new one. I've switched out a base. So in this case, I've changed the sequence of DNA. Now, if you remember, a change in DNA really essentially means a change in the amino acid sequence. Different amino acids, very different properties. Let's go into them. Alright, so the first one we're going to go into is substitution. This is when you have a portion of the DNA replaced or substituted by a different base pair. Here's my original strand of DNA. You can see one of the letters has been substituted or changed. The A was replaced with a G. So I like to use this example. It's like codons. All this stuff is three-letter words. The fat cat ate the wee rat. In this case, a substitution is going to change it. Now in this case right here, the C has been substituted for an H. So instead of the fat cat ate the wee rat, we have the fat hat ate the wee rat. Substitution changes the meaning of the sentence, but for the most part, it's still readable. Alright, I want to show you our normal DNA strand, and I can't stress this again, this is different than the one you have in your packet. So I'm going to be first transcribing that DNA to RNA. Remember where you would have normally had a T, now you replace that with a U. So I've transcribed that all into RNA. Now I'm going to translate that into protein. AUG gives me methionine, my start codon. CAA is going to give me glutamine. Next up, CCC gives me proline, PRO. AAG is going to give me lysine, LYS. UGU gives me cysteine, and UAA gives me stop. Okay, so let's take a look at our substitution mutation. Remember, different sequence than what you have. On yours, make sure that you circle any DNA bases that have been mutated or changed. That T was substituted for a C, that A was substituted for a T. So that original strand of DNA has changed. Now I'm going to transcribe that into mRNA. Remember, A with T, normally we would have had a T inside the RNA, switch that out for a U. So this one here I'm completely transcribing. It's going to be pretty much the same as what you've seen before. Speed it up a little bit on the bottom here. Remember, everything's the same except for that one mutated base there. Now I go back and I start translating that into protein with thionine. Remember, this one has changed. There was a mutation, but it actually didn't change the amino acid. CCC, same before as proline. AAG is going to give me lysine, cysteine, and stop. Bottom pretty much going to all be the same until I get to that fifth one over there. That one is going to be serine. Remember before it was cysteine, and finally once again to stop. Now you want to circle any bases that or any amino acids that have changed. In this case, it's just the one. Cysteine was switched for serine, but this one, even though there was a mutation there, did not result in a different amino acid. That is what is called a silent mutation. Alright, now let's try an inversion one. Inversion is essentially when a part of the DNA is inverted or switched around in the chromosome. Here's my original strand of DNA. Here it is with part of it inverted. It's kind of hard to see, but if I take the CAT and flip that part around, that gives me TAC. So that one little fragment flipped around, just like, I think, Bobby's name from Family Guy. Well, an inversion takes place right there. Sometimes this can be an inversion all the way on the chromosomal level, where the chromosome will pop out and flip around depending upon where it is. Or in our case, we're going to take our sentence and we're going to flip it around. So instead of the fat cat ate the wee rat, it's now the fat tar you cat attacked. Now that's somewhat illegible right there, but it's because I've taken this whole part here and actually switched it around. So it's the fat cat ate the wee rat, but backwards. In this case right here, that sentence is now almost completely unreadable. Let's try an example. Alright, the next one I'm going to show you is a rearrangement or inversion mutation. So I've already drawn a box around the parts that's actually switched around. I'm transcribing that all into mRNA right now. Remember, everything's going to be the same except for those areas that are in a box. Now I'm going to translate that methionine. This one's going to be a little bit different. It's glutamate instead of glutamine. Proline, asparagine, cysteine, stop, those haven't changed. Methionine, glutamine, proline, lysine, cysteine, and stop. Now, in this case, if you circle any ones that have changed, I only have that one and the asparagine that has changed. Proline did not actually change because when you switch around CCC, you get CCC. There has been no change there. On the bottom here, you also have what is called a palindromic sequence. This one did not change because ACA backwards is ACA. Alright, now let's try an insertion one. Essentially, this is essentially what it sounds like. When you insert something into that original strand of DNA, TAG, CAT, if I insert something in there, I've inserted the G. It shifts everything else down a little bit there. That extra G was inserted. Let's try our example with the fat cat. So the fat cat ate the wee rat becomes the fat cat ate the wee rat. Now it's that little in the middle there that is new. That is something else. Meaning is still mostly clear. Deletion, pretty much the same, the other side of the coin here. When I have part of the DNA deleted or removed. So here's my original, here's my new one. As you can see, the AT fragment was deleted. Sometimes this actually happens at a giant chromosomal level. Something like there's a genetic disorder called cri du chat, or cry of the cat, where part of it is removed. Click right here if you want some more information on that. So, back to our example, this originally was the fat cat ate the wee rat, now it has become the cat ate the wee rat. We have removed the word fat. This actually does change the meaning of the sentence. Sometimes a deletion can be catastrophic, sometimes not so much, you can still get the gist of it. Let's try our examples. Alright, now I'm going to go through an insertion and deletion. Sorry about my bad spelling there. Draw a box around any bases that have been added. In this case, I've added those ones and removed those ones here. So I'm going to transcribe those into mRNA, same as before. I have to add in that additional one here. Here I don't have to account for that one because that was deleted. Now I'm going to translate those into proteins, into my amino acids. You know, nothing new here. This one is a new addition. Glycine is new. The rest of those are the same. Obviously, I have skipped out on the glutamine that has been removed. So if I take a look at what's been added or removed, my glycine was a substitution, or sorry, an insertion. And right here, my glutamine was actually removed. You normally would have had it right there between the methionine and the proline, but in this case, it is gone. All right, our last one right here, frame shift. Sounds weird, is a little bit different, and it can be the most catastrophic depending upon where things go. In this case, I'm going to delete the letter T. Not the word, just the whole thing. So the fat cat ate the wee rat becomes the fat cat tet hew ear ate. Now, the whole problem here is everything got shifted down. The reading frame was shifted. It originally was the fat cat and then ate the wee rat, and then this other letter popped in from the side there. But just a deletion of a single letter messes up everything else. So not only did it completely change the meaning of the sentence, that one word changed and every other word after it was also changed. It can be a catastrophic mutation because a single letter added or deleted will screw everything up. On to our example. All right, now we're going to look at frame shift, which is probably the most catastrophic, but you might not think it right away because it seems to be the smallest addition here. So I'm going to take my sequence, and remember, the C has been deleted. Everything after it shifted down to compensate. Right here, those two A's have also been deleted. Everything else shifted down. So now, when I'm translating these into proteins, everything is moved down one and it's shifted over. It's almost illegible. Isoleucine, asparagine, proline to serine, to valine. We haven't seen some of these before. To asparagine again, methionine. See, right down here, it's mostly okay because this is where the shift is. Now, this stop actually is kind of important right here because remember, this is the period at the end of a sentence. And in this case right here, it doesn't really matter what I have after that. That wouldn't be translated anyways because it would get to this and it would stop. So when I deleted that thing, I'm actually deleting an entire base there. So if I'm to circle all the ones that are different, isoleucine, asparagine, serine is new, valine is new, another asparagine in there. One tiny letter caused 1, 2, 3, 4, 5 differences. Down here, the actually removal of twice the amount only caused essentially 2 new changes, even though essentially what it did was it jammed in a period into here where there was not before. So that threonine would never have been translated in the first place just because that stop codon was created.